which is on photophosphorylation. Now, you have already learned about the oxidative phosphorylation. Today, we are going to talk about another mode of phosphorylation by which ATP is being synthesized. And here, what you will see is that the source of energy is different than what you have learned in oxidative phosphorylation. What you have learned in oxidative phosphorylation is that you have a fuel molecule like glucose. Glucose is broken down to a simpler molecule and then is getting oxidized completely into carbon dioxide and water. And in turn, what you get is you get the NADH and ATP, and then this NADH by oxidative phosphorylation is converted into ATP. So that is what you have learned so far. Now today we are going to talk about the mechanism of ATP synthesis. You will see a lot of similarity, some differences. So that is what we are going to talk about. The purpose of today's class is that you should also learn that there are some other way or mechanism by which ATP can be synthesized. As well as you will also learn that during this process where this oxidative phosphorylation can produce ATP, there is also photophosphorylation where you will see the synthesis of NADPH. Now here, when you are learning about the synthesis of NADPH, it is a different thing. So far, we have talked about NADH. Now we are going to talk about NADPH. As I discussed when we were talking about the vitamins and coenzymes, that NADH and NADPH are more or less same time reduced molecules. Hello? Okay. Can you hear me?
Yes, sir.
Okay, fine. So what I'm discussing here is that as we discussed earlier, you have a molecule NADPH. Now, as you know, that the presence of
Sir, your
this
voice is not.
phosphoryl group over here. Am I audible?
Sir, now you
Can
are,
you hear
but
me?
yes, sir. But
Okay.
before that, for
Okay.
like a
So
minute, your
let
voice was.
me. It is probably the distance of the lap. No, my what here, what you have is NADPH, and what you get in case of the process where we go for citric acid cycle, you get NADH. So the difference between NADH and NADPH is only one phosphate residue which is present in the molecule. Now, this presence of phosphate tells that this system is energy rich. So wherever you have NADPH, this NADPH is the one which will go for biosynthetic reaction. This part you will also learn in other cycle where NADPH, when it is being present, it is going for the anabolic reaction where it will synthesize the molecule. Whereas if NADH is there, they will go for catabolic reaction where they will be responsible for the electron transport chain and production of ATP. So what important thing in the light reaction is that carbon dioxide and water, they are making carbohydrate. So far you have learned about how carbohydrate can be degraded, how the bond energy can be utilized for synthesis of another bond energy in case of in the formation of ATP. Now here what you are learning is that the light energy is being utilized to synthesize bond energy which is present in the carbohydrate. Now the process as conventionally written, so the first equation which is written where CO2 plus H2O in presence of light is making CH2O, that is a carbohydrate plus oxygen. So there is a process where oxygen evolution is occurring. You are learning that in presence of light, this is being synthesized. So actually, people, they have differentiated that it is not the light which is required for carbohydrate synthesis, rather it is the product of the light, that means synthesis of this NADPH and ATP, which is driving the synthesis of carbohydrate. So even if you can carry out the reaction of the synthesis in presence of if I have NADPH, if I have ATP, I can synthesize CH2O, that means carbohydrate, even in absence of light. So what I mean to say here is that actually for synthesis of carbohydrate, light is not required. The product of the light, that these two products, NADPH and ATP, is required. That is why you require light. So energy has been trapped in the form of NADPH and ATP. So we will be learning how this is being done. Another thing you will learn is that in case of oxidative phosphorylation, most of the processes were happening inside the mitochondria. You have a glycolysis from where the NADH or ATP is produced, but that ATP is a very, very small quantity. The maximum ATP comes when NADH enters inside the mitochondria. So I have not drawn mitochondria. I'll show you at the end that the mitochondria is the one which is a powerhouse. Most of the ATP which is being produced is inside the mitochondria. And how it happens? Because citric acid cycle is happening inside the mitochondria, the NADH is being produced inside the mitochondria, and inside the mitochondria, in their inner mitochondrial membrane, your electron transport chain happens, as well as your oxidative phosphorylation happens, and because of that, ATP is being synthesized inside the mitochondria. So that part you have already learned. Here, a similar kind of a structure is there, but not exactly same. Here you have chloroplast. Chloroplast will have grana and stroma. Grana is the stack of these, thylakoid membranes. So these are thylakoid, which are stacked one above the other. And then the matrix present over here, the grayish color, is the one which is called as stroma. So you have a stroma and grana, and of course you have inner membrane, outer membrane of chloroplast, and everything is happening in these membranous structure. So you will be learning how this process is happening. So actually what happens, this kind of a reaction you have learned where QH2 or quinol synthesis occurs. Here we call it the plastoquinol because it's thought to be present in chloroplast or plastids, and that's why it has been referred as a plastoquinol. So the plastoquinol is the one which produced by photosystem 2. Don't get worried, I'm just going to talk about photosystem 2. So photosystem 2 is here. The photosystem 2 is the one which accepts light or photon or energy from the light at 680 nm. So you will have
Sir,
the
we are still on the light reactions of photosynthesis slide with the stroma, lamellae, and the inner cell
uh,
first.
I do not know. Let me just check it. My should be. So can you see slide which is showing cytochrome BF link photosystem 2 to photosystem 1?
No, sir.
Okay, let me go back and then again re-enter. So what slide is visible to you?
Yes, sir. Now we can see cytochrome BF links.
Okay, so this slide explains that how photosystem 2, which is written as a PS2, you can go on the left hand side bottom. I have shown you that there are two photosystem. One is a photosystem 2 and another in yellow color, which is a photosystem 1. So photosystem 2 is accepting light, and the one which is accepting light is at 680 nm light is being accepted. So you have a different photosystem which is accepting light. Here it is 680, where here you will find that it is at 700 nm. So these two are differentiated only based on their ability to accept the light or photons. So here The photons, they are exciting at the PS2. I'll give you the complete cycle, but this is the one which explains how QH2 is going to be converted into Q. And so the simple mechanism is that here the photons, they come, they excite, and after excitement, the electron is being, so you will get a hole of electron in PS2. And how it is being filled? It is being filled by this splitting of water, which will be converted into oxygen. So this is the process by which oxygen evolution occurs in photosynthesis. And then what will happen? The electron will move, it will go to photosystem 1. And in the photosystem 1, the main process is that NADP plus is converted into NADPH. So here you get the potential which is in the form of NADPH. And in the mean way, in intermediate steps, what you get is that quinol is going to be converted into quinone, and this process is occurring by a plastocyanin. This is a copper protein which is present in thylakoid lumen. So my major concern is that how the electrons are being, the energy is being trapped from photons, and that is what we are going to explain. There are some intermediate steps which is important. For example, ferredoxin is another very important component of this photosystem 1. And what happens that chlorophyll A lies in the center of the structure and absorbs light. Maximum absorption is being at 700 nm, so that is why it is known as a P700, and it initiates photo-induced charge separation. So you will be learning that how these photons, they are helping in the whole process. And other than that, the electrons are transferred down a pathway through chlorophyll, and this is a site A0 and quinone at a site A1. So these are not very important because these are the integrities, how the electrons are being moving from one complex to another complex. But let me just come to this. This is a very interesting and important scheme. It is known as a Z scheme, and this explains the complete system. And this is known as acyclic phosphorylation. Here, the electron flows from, first is that here light energy will come. Let me just show it to you. So light will come and excite this P680, and when it excites the electron from here, it will go to the excited state. So that is why this is called as a P680 star. That means it is in the excited state. Now from here, the electron will go, and whenever we are talking about going down, it is based on the reduction potential of each molecule here. For example, pheophytine and other quinol, then cytochrome B F complex. So this whole structure, whole scheme has been arranged based on their reduction potential. And we have talked about reduction potential just yesterday. So that means how these molecules, this gives you an idea that what is the kind of electrons which can accept and how they should be present in a cycle. So for example, if you have more reduced, more negative charge here at P680, it can move to lesser negative charge at pheophytine. So that's why electron moves from here. And same way, cytochrome BF complex will be having much lesser reduction potential. That means this will be more towards the positive charge. So that way, it has been completely arranged. So ultimately, what you see is that the electrons which are moving from P680 to excited state of P680, which is the photosystem 2, the electron which has been, here, once the electron moves, you create a hole. And this can be, your electron is coming at this point by the splitting of water. So this water will split into oxygen, and the one of the electron will flow over here. Now, this way, your electron keeps on flowing from pheophytine QA, Q2 to cytochrome BF complex. And this complex I will talk about in detail. This is a very important complex. So there are two important complexes here. One is cytochrome BF complex, and another is FD-NADP reductase. So the cytochrome BF complex is the one where ATP is being synthesized, and FD-NADP reductase is the one where NADP is converted into NADPH. Now, this kind of reaction is possible when we called about the cyclic phosphorylation. There is one more interesting point we'll discuss, where ferredoxine, if NADPH is NADP plus is not present in sufficient quantity, then electron will flow from ferredoxine to cytochrome BF complex. So if I say the electron is flowing from here to cytochrome BF complex, that will be called as a cyclic photophosphorylation. So here we are talking about the electron flow, which is a non-cyclic photophosphorylation. Another type we will talk about, where ferredoxine is giving its electron to cytochrome BF complex. And if so when this cyclic photophosphorylation will be possible? Only when you have less amount of NADP plus. That means most of the NADP has already been converted into NADPH. So it's the ratio of NADP plus and NADPH which decides whether it should be cyclic phosphorylation or non-cyclic phosphorylation. Now, as I said in the previous slide, that this ferredoxine NADP plus reductase is the one which is responsible for converting your NADP plus to NADPH2. So this reductase complex, it is a, this is an enzyme which is known as a ferredoxine NADP plus reductase, and this enzyme will have a binding site for ferredoxine where the electron can move. Then you have NADP plus binding site. So these two are substrate, and then these substrate, they will be helping the electron to flow. So you have this ferredoxine NADP reductase, where the electron comes, it will reduced form will be oxidized, and you get FADH. Here it is in the form of semiquinone. So here you have just electron present in FADH plus FADH, and this is called as a semiquinone intermediate. This semiquinone intermediate can be converted into FADH2. And now I have talked about FADH2. Tell me how the electron will move from FADH2 in case, if I talk about oxidative phosphorylation. In case of oxidative phosphorylation, you have succinate dehydrogenase, and in the succinate dehydrogenase, the electron will be flowing from complex number 2 to complex number 3. But here you do not have those complexes. Here you have a different mechanism where ATP is being produced. So what I'm talking about is there is a lot of similarity between the oxidative phosphorylation and photophosphorylation. We are going to talk about, so here the mechanism is that you also have a proton gradient, like you have seen the proton gradient over here. So in oxidative phosphorylation, what is happening? A proton gradient was generated because the electron was flowing in the electron transport chain, and from each site, each major enzyme, you will finding that the protons are being pumped out. And because of the pumping out of the proton, when the proton re-entered inside the mitochondria, what you have got is that you got ATP synthesized. Now here, again, the proton gradient across the thylakoid membrane is responsible for ATP synthesis. The only mechanism which is differing between oxidative phosphorylation and here is that in oxidative phosphorylation, the electron transport chain is responsible for the gradient. Here, what you learned is that this pH gradient is created and in the thylakoid space. So the first thing is the sites are different in oxidative phosphorylation and photophosphorylation. Second thing is that the mechanism of electron transfer is different. Here you see that the light reaction and the cyclic phosphorylation and acyclic or non-cyclic photophosphorylation is responsible for flow of electron. So what we are discussing in this one is that the pH difference which is being created. So light induced transmembrane proton gradient is being created. So when you have the light exposure, just go back to this slide. Here, what is happening? There is light which is exciting P680, and then electron is flowing, and then it is going to NADPH. Now, during this process of electron flow, the transmembrane proton gradient is being created, which is around 3.5 pH. And this 3.5 pH, if I calculate, it will come out to be minus 4.8 kilocalorie per mole. So this pH gradient is the one which is going to synthesize your ATP. And the difference, what you find here is because that thylakoid membrane is quite permeable to chloride and magnesium ion, and that's why they are, that is why you find that this difference occurs between the oxidative phosphorylation and photophosphorylation. Now, coming to the ATP synthesis, I don't have to explain much because you already know the story that when proton re-enters inside the mitochondria, then it is happening. The only difference is that here the ATP synthase mechanism is slightly different. It is the orientation is different. So that is the part I'm going to show it to you. So what will happen that the proton motive force generated by the light reaction is converted into ATP by ATP synthase, which are present in chloroplast. And here the complex is known as CF1 and CFO. So CF1 and CFO is just nothing but similar complex which you have learned yesterday about the F1 F0 complex in mitochondria. The CFO conducts proton across the thylakoid membrane. You have learned that FO is conducting the transport of electron in oxidative phosphorylation across the mitochondria. So this is the proton channel. CF0 is proton channel, and CF1 is the one which is a catalyst, which will be leading to the formation of ATP from ADP and PI. The significantly membrane orientation of CF1 and CF0 is reversed compared with the mitochondrial ATPase synthesis. In mitochondria, what happens that F1 part is on towards the mitochondrial matrix and F0 part is outside the mitochondria. So what you are learning here is that in this case, you will see that from the grana, how the orientation occurs, it is the reverse one. Proton flows out of the thylakoid lumen. So thylakoid lumen, you have seen here, the membrane is present on the inner surface, which is called as the thylakoid lumen, and where your synthesis of ATP is being happening, and NADPH formed through the action of photosystem 1 and ferredoxin NADPH P plus reductase also is released into stromal space. So let me just go back to show you stromal space. So here you have a stroma. Here you are, so most of the process is occurring in thylakoid membrane. So everything is happening here in thylakoid membrane. In case of oxidative phosphorylation, you have seen that everything was happening in inner mitochondrial membrane. So if I say that mitochondria, the ATPase is present, so ATP synthesis will be happening in stromal part. So that's the way it is different than the your oxidative phosphorylation. Okay, so this is the figure which will show you how the photosynthesis is different than oxidative phosphorylation. Though both are the mechanism where in both of the cases phosphorylation occurs, so it is not exactly photosynthesis. I'm just focusing on the photophosphorylation. So the photophosphorylation and oxidative phosphorylation, they are different in the sense that one is happening in mitochondria and this is happening in the chloroplast or in thylakoid membrane. So what you are learning here is that the proton is pumped in, and in oxidative phosphorylation, what you see is that in both the cases, what you are learning is that here, in case of photosynthesis or photophosphorylation, synthesis of NADPH is an important event. Here, in oxidative phosphorylation, the conversion of NADH to NAD plus is an important event. So here, it is leading to synthesis of a molecule which can in future synthesize other important molecule. That means NADPH is going to lead to the biosynthetic reaction. Here what happens that whatever you have gained NADH by your TCA cycle, that is going to be utilized, converted into NAD plus, and the energy trapped here or redox potential of this will be converted into phosphoryl group transfer potential. So these are some important differences. If you look at the orientation, here ATPase is oriented in such a way that it is going ATP synthesis occurring inside the mitochondria. Here it is it's a reverse process where protons are pumped out from. So let me just go back again to explain this. So here, if I say that they are in the stromal membrane and they are synthesized, still they are synthesized in the granule part. So sorry, there will be, so if this is a grana and it is being synthesized, so if ATP is oriented on the outer side the way it is shown here, so if I say here, so ATP is synthesized here, this ATP will be present in the inside the your chloroplast but in stroma. Now, coming to the cyclic electron flow, I have already shown you that how cyclic electron flow can occur. If your NADP is in excess quantity, then cytochrome B complex will receive electron from, from the enzyme. So if you look at this one, if I have excess of NADPH, that means energy is enough, or your the phosphorylated molecule in reduced state is enough, that means you need more ATP. Or if you do not require NADPH, you just require ATP, so the electron will flow from ferredoxine here. Electron will flow from here to cytochrome BF complex. So from here to here. And this flow will generate ATP when it is moving in cytochrome BF complex. So these are the things which are happening in case of cyclic photophosphorylation. So yeah, so you have learned that where this synthesis occurs, how the light reactions they are helping in the transfer of electrons. You have also learned that how ATP is being synthesized. At present, we are talking about cyclic electron flow. So cyclic electron flow is the one where electron is reduced ferredoxin. In reduced ferredoxin can be transferred to cytochrome BF complex that you have seen, and this can happen um, because the electron is not going to NADP plus. So it is not going to reduce NADP plus into NADPH. So in this case, you will be getting your no NADPH is being produced, rather ATP is being synthesized. This electron flow back through the cytochrome BF complex to reduce plastocyanine, which can be reoxidized to P700 to complete a cycle. So actually, to explain, I have to again go back to show it to you. So suppose what will happen, cytocomplex plastocyanine to P700. So it is come back. So cyclic nature is because ferredoxin gives its electron to cytochrome B complex, then goes to PC to P700, and again. So it becomes a kind of a cyclic phosphorylation. Now we are, just let me. So you have seen all these things. We have to switch to cyclic electron flow. So you have learned how cyclic electron happens. Electron flows back through the cytochrome BF complex, reduce PC, which can then be reoxidized by P700 plus to complete a cycle. The net outcome of cyclic flow of electron is a pumping of proton by cytochrome BF complex, and the resultant proton gradient, as I said, you will create a proton gradient, and this proton gradient will synthesize ATP. In this process, it is called as a cyclic. As you have seen, it is a nature that it is not acyclic or non-cyclic. It is a cyclic photophosphorylation. ATP is generated without concomitant formation of NADPH. That means you are not going to convert them into the form of NADPH. Rather, it will be NADP plus will remain as such because you already have high level of NADPH, and that's where the electron will flow back to cytochrome BF complex. This is showing you the mechanism by which ATP is being synthesized. So your PC is the one which is in the oxidized form. It will become in reduced form. During this process, proton will enter, and in turn, this is the one where your excitation occurs. And what is happening is that proton, when it is flowing back, and this ATP is being synthesized. So this way, you have learned that how non-cyclic and cyclic phosphorylation they work in the the photophosphorylation process. Now, let me know if you have any question. Hello? Do you have any question?
No, sir.
Okay. So here, I do not want to give a lot of emphasis on photophosphorylation, except that my purpose is to explain to you that in case of photosynthetic bacteria, in case of plants, the mechanism of ATP synthesis is slightly different, but the fundamental remains same, where electron flow is the one which is responsible. Of course, the electron flow are in a different through different intermediates, and this electron flow ultimately gives you the proton gradient, and this proton gradient is the one which is responsible for the synthesis of ATP when the proton flows back from one compartment to another compartment. Of course, the orientation may be different, of course, the organelle may be different, but the principle remains same. So suppose yesterday we have talked about that ATP is being produced from the energy which is generated because of the electrochemical or PMF, proton motive force. So proton motive force generated across the mitochondrial membrane produces ATP. Same way you can say that proton motive force which is generated across the chloroplast membrane, which is thylakoid, that is the one which is responsible for producing the ATP molecule. So both are same in terms of the mechanism. Other mechanism which I have told was that when proton flows back during this process, the structure, the binding change mechanism happens. As soon as proton binds and enters inside the mitochondria, what you see is that ATPase changes its structure. It is a loose form, tight form, open form. So at first it binds at one binding site, when then binding site changes from one state to another state. So when it changes to tight form, the ADP and PI will come close to each other, and then in the next form what will happen, ATP is being synthesized and they will be released. So this way you have learned the ATP synthetic mechanism, both utilizing energy from fuel molecule like carbohydrate and from photons, which is in case of photosynthesis. Is it clear? See, you have to go through this process, and if you have any difficulty in understanding, please let me know. What I'm doing is that because your course is not about the photosynthesis as such, is about just learning about photophosphorylation, I am trying to focus on photophosphorylation alone. Now, what we are going to do is that we are going to discuss, once we are through with this, let us talk about something uh, different. And here we are going to talk about a pathway which so far you have not discussed. I thought this is important pathway because this ultimately will be the one which is going to synthesize carbohydrate, as well as here in case of human beings, it is an important pathway where it supplies a lot of important carbohydrate. And this pathway is known as the pentose phosphate pathway. Have you learned this pathway in microbiology?
Yes, sir.
Yes. Okay. So then it is my job is simple. I will just go through some of the
So, but
processes
in a
which
very
may
brief
be
manner. Very brief manner.
okay. But anyway, here you are not required to learn every steps. But what you are important is that you should know why this should happen. What are the different phases of pentose phosphate? So when we are going to talk about pentose phosphate pathway or HMP shunt, it is going to talk about two steps. One is a non-oxidative phase, where there is no oxidation happens, and it catalyzes the interconversion of different carbohydrates. That means 3 carbon, 4 carbon, 5 carbon, 6 carbon, 7 carbon. So what you have is that this pathway, different sugar molecules, they can change from one form to another form. Suppose you have 6 carbon, if you want to convert them into 5, this is the say 6 phosphate is converting to ribose 5 phosphate. What does it mean? It means things. Where do you require ribose 5 phosphate? You require ribose 5 phosphate for the nucleosynthesis, but ribose is the one which is required. You require this for many coenzyme synthesis. You require it for several other processes. Other than that, you also get NADPH. NADPH is nothing but redox potential. This is the potential which will be required for other biosynthetic processes. So the conversion of this, or the first reaction of the pentose phosphate pathway, which is leading to many other pathways. So what I'm trying to emphasize here is that is a quite important pathway which is responsible for different reactions. Now, so to summarize, the first reaction is glucose 6-phosphate. From where you get glucose 6-phosphate? This you get from anyway in glycolytic pathway, isn't it? In glycolysis, what you see is that glucose 6-phosphate is converted into glucose is converted into glucose 6-phosphate by hexokinase. Do you remember that? The first step in glycolysis. Now, from glucose 6-phosphate, if it goes to fructose 6-phosphate, that means it is going towards the glycolysis. If glucose 6-phosphate is converted into ribose 5-phosphate, that means it is going for the pentose phosphate pathway. So glucose 6-phosphate is the one where your diversion can occur. You will also learn that there are different other pathways which can lead from glucose 6-phosphate. And that is what was the reason we have discussed that hexokinase is not inhibited or is not the one which is regulating glycolysis in general. It is the fructose phosphofructokinase which is regulatory regulation, which leads to regulation in glycolysis. Do you recall this? Okay, let us move on. The pathway, which is taken from Schreier's book, phosphate will lead to the ribulose 5-phosphate and then ribose 5-phosphate xylulose. Going to talk about all these things. Carbon. So this is the oxidative phosphate pathway. So your 6 carbon is converting to 5 carbon, and this NADP plus, which is present in the system, is being reduced the oxidative phase, which is going to generate NADP. You have a non in the, what it will do is it will convert phosphorylated sugars. So you will have fructose 6-phosphate, it will convert into xylulose 5-phosphate, fructose 6-phosphate, pseudoheptulose. So all these reactions which are shown below this, they are coming under non-oxidative pathway. Now, so what will happen? What is the end result? End result is that you are getting 2 molecules of NADPH which are generated in conversion of glucose 6-phosphate to ribulose 5-phosphate. Now, the oxidative phase of pentose phosphate pathway starts with the dehydrogenation of glucose 6-phosphate, a reaction catalyzed by glucose 6-phosphate dehydrogenase. You have learned wherever NADPH is involved, or wherever NADP is involved, or NAD is involved, all those places you have dehydrogenase. You have learned this in case of your citric acid cycle, in case of your glycolysis. So dehydrogenase's job is oxidation reduction. The product what you get is 6-phosphogluconolactone, which is intermolecular. I will show you this reaction. The next step is the hydrolysis of this lactone in the presence of lactonase, and you get 6-phosphogluconate. So this is a kind of intermolecular change, and that's why the way you get 6-phosphogluconate, and which will be, so this is so far it is 6 carbon. Then oxidatively it will be decarboxylated, and this is the same thing you have learned in case of your citric acid cycle also, where you have seen the isocitrate dehydrogenase. It's oxidizing as well as decarboxylating. So here also decarboxylation will occur. Why decarboxylation occurs? Because it has to remove one of the carbon from the ring or from the structure, and that is where it is happening. So 6 carbon sugar is then converted into 5 carbon sugar, which is a ribulose 5 phosphate. NADP plus is again the electron acceptor. The final step of the synthesis of ribulose 5 phosphate is isomerization of ribulose 5 phosphate. Here you have enzyme glucose 6 phosphate dehydrogenase. NADP will be required, and what it will do is that it will produce one molecule of NADPH. So one molecule is being produced. It is converted into lactone structure, so it is a ring structure. From ring structure, it is again converted into open structure in presence of enzyme lactamase, and lactamase is converting them into a form. So what is happening here, from this to this and this to this, what you get is that one carbon dioxide is made in such a form that it can be lost. So in the next step, what you get is that 6-phosphogluconate, where one carbon dioxide is released, as well as you get ribulose 5 phosphate. Good thing is that here you started with the compound which was a glucose 6 phosphate, which is aldose, and here also you are getting an aldose. But then they can be isomerized, they can be converted, interconverted into other forms where they can be form, conversion occurs. So this is the reaction. Okay, sorry, I moved on. So this reaction shows you the oxidation reduction reaction, where glucose 6 phosphate is converted into ribulose 5 phosphate. Now, next one is where there is no oxidation reduction reaction. So there are two enzymes. One is a transketolase, another is a transaldolase. And what you find is their very unique mechanism. Transketolase will take two 5 carbon compound and will make one 3 carbon and one 7 carbon. Then another reaction catalyzed by transketolase is C3 plus C7, the one which is the product of the first reaction, and it will form C6 and C4. Ultimately, you get C. And here, if I take C4 from here, this one, and C5 from first one, you get C4 plus C5, which is making C6 plus C3. See, if I sum up all the three reactions, what you find is that 2C5 here and 1C5 here. So you have 3C. And here, what you have is 2C6. This one and this one, 2C6, and one C3, and others are being utilized in the reaction. For example, this C3 produced in the first reaction, the reaction is utilized in the. So then results.
Sir, your voice is cutting.
This is the reaction which is shown here. Yeah.
Sir, your voice is
Hello?
cutting out. Your voice is cutting out.
Okay, so what I'm saying is that you have 2C5, which is going to make you C3 plus C7. And that is where you get, as I have shown you in the previous slide, this one, that first reaction is 2C5 giving you 1C3 and 1C7. So what is the reaction? You have xylulose 5-phosphate, ribose 5-phosphate giving glyceraldehyde 3-phosphate, and pseudoheptulose. Pseudoheptulose is 7 and it is 3 carbon. Now the next reaction is the one where you have glyceraldehyde 3-phosphate and pseudoheptulose 7-phosphate, the product of the previous reaction, and that you get is the fructose. So I'm just increasing the size. You have fructose 6-phosphate and erythrose 4-phosphate. Fructose 6-phosphate is 6 carbon, it is a 4 carbon. So you got the second reaction. And third reaction is where one of the that is erythrose 4-phosphate, and one of the beginning one, xylulose 5-phosphate. So one 4 carbon and one 5 carbon in presence of transketolase will give you one 6 carbon and one 3 carbon compound. So this way, what you have learned is that utilization of a 3-5 carbon compound is giving you the product. So at the sum, what you are getting is that 2 5 carbon compound are 2 xylulose 5-phosphate and 1 ribose 5-phosphate. So these are 2 5 carbon compound, and 2 6 carbon compound is fructose 6-phosphate, and 1 3 carbon compound is glyceraldehyde 3-phosphate. So this is the net reaction which is happening because of the 3 other reactions, because some of the metabolites they are being utilized, and ultimately what you produce is that from 3 5 carbon compound you are producing 2 6 carbon compound and 1 3 carbon compound. So sum of the reaction is the same. And what you also see is that ultimately it is starting with 3 ribose 5-phosphate. What are the 3 ribose 5-phosphate? Which has been made from glucose 6 phosphate, which has been converted into ribose 5-phosphate, and then the reaction starts. So I can say that these 3 ribose 5-phosphate are the one which are being converted into 2 5 xylulose 5-phosphate, and one remains as such. So at the end of the reaction, what you get is that 2 6 carbon compound, that is 2 fructose 6-phosphate and 1 glyceraldehyde 3-phosphate. So if I sum up all the reaction oxidative pathway, it is glucose 6-phosphate giving you 6-phosphogluconate NADPH. This I have already discussed. All the reaction I have discussed, just it is a summary of the reaction, what is coming under oxidative phase, what is coming under non-oxidative phase of your pentose phosphate pathway. There are flow. Now you must be wondering that how the flow is being generated. So depending on glycolysis and pentose phosphate pathway, the flow is decided whether glucose should go for the further conversion to pyruvate, or glucose should enter into the pentose phosphate pathway. And this kind of a mechanism will depend on different modes. So there are 3-4 modes which have been given as been described. First mode is where much more ribose 5-phosphate than NADPH is required. So this is the requirement of the cell. So glucose 5 glucose 6-phosphate plus ATP, this will give you 6 ribose 5-phosphate plus ADP and proton. What it means is that you are creating a lot of ribose 5-phosphate. When do you think this process should happen? This can happen when you have very fast nucleic acid biosynthesis. You require a lot of ribose for synthesis, then too much of energy ribose. So this reaction is a preferred one. The second one is where the need of NADPH and ribose 5-phosphate is a balance, which is a common, which is the normal cycle, which is given as I described earlier. That means you will be producing 1 molecule of ribose 5-phosphate and 2 molecules of NADPH. Same way, third is where much more NADPH than ribose 5-phosphate is required. So you see the reaction. So it is the cycle pentose phosphate pathway as well as the glycolysis, they merge with each other to show how the flow of the cycle should occur. And this is the fourth and the last one, I think. So this is the last one, which is showing you that how glucose 6-phosphate can be converted. And here, both NADPH and ATP is required. So both of them are required. So under those conditions, this kind of reaction can occur. And this is the one which shows you under different mode how the cycle converts from on each other. Maybe in the next class, because today time is not there, I will take few minutes to describe it, how the balance occurs. So on the left-hand side of each cycle, I have shown you the glycolysis. On the right-hand side, it is the pentose phosphate pathway. So there is an interplay between pentose phosphate pathway and glycolysis, by which different mode the reaction occurs. So by this, I have completed this chapter. You have already learned it. Just it is a refresher and making you to understand how the different pathway can enter from glycolysis to pentose phosphate pathway or vice versa. So if you have a question, probably today we don't have time. In the next class, you can ask. Okay, then I will sign off.
Sir,
Thank you very much.
sir, excuse me, sir. Sir,
Yeah.
sir, I
Yes,
had
I
a
can hear.
question.
Yes.
Sir, the graphical abstract.
Right.
So, sir, how, sir, so can we make it by hand or like do we have to make it on
See,
some
as you will. You can make it by hand and make a copy and give it to me. And
Okay, sir.
alternatively, if you can make with some, PowerPoint or some graphical tool, you can make that also.
Okay, sir.
That's
Sir,
not
one
a
more.
problem.
Sir, one more question. Sir, you said 4 to 5 pages, sir. Sir, but sir,
Yeah.
if I make 4 to 5 pages, if I do it by typing, then it would be like 2,500 words. But if I do it on paper, it would be like around 500. So, sir, if you could give us a word limit, then we would be better able to like get the size of our. Uh, mm,
So if I
site.
say one page is equivalent to something like 200 words,
Yes, sir.
5 pages, 1,000 words.
Yes,
So
sir.
it comes out to 1,000 words. I will just mail it so that everybody knows it.
Yes, sir.
Um, so 1,000 word is where you can, very nicely write. I don't need too much of writing. I want 4 to 5 pages. I have converted this from handwritten. So
Okay,
let me give you a range, 1,000 to 2,000 words,
okay, sir.
because some people they may write. So I'll give you the range where you can write between 1,000 to 2,000 words, and it is up to you. If you want, writing concisely is much more difficult than writing blah blah blah. So
Yes, sir.
one can, you know, do it. I will just mail it to all of you for your information based on your query.
Okay. Sir, if we are typing it, can we add pictures?
See, don't copy and paste. That is not required.
Okay, sir.
You can add whatever you want, but then don't copy from the journal paper and paste it, because that has no meaning. That is already given. I need some input from your side. And if it is your own, then it makes a sense. Otherwise, if I take a copy, it is just copy paste thing, which will not help. Okay. Any more questions?
Sir, and sir, about the major syllabus, sir.
Syllabus is whole paper, whole chapter, means full course.
Okay, sir.
Because major, and because it is open book, so it doesn't matter. You are not cramming anything.
Yes.
Everything is from the book, and everything has been, and everything is going to be whatever has been taught. So you are not, going beyond a certain thing which has already been taught to you.
Okay, sir. So, sir, today the syllabus was completed. So on Friday we will have the
No,
quiz.
it is not completed. Where is it completed? Still you have, you look at your things. I still have to cover a couple of things. For example, I have to cover the glycogen metabolism, fatty acid oxidation, signal transduction. A lot of things are left in the syllabus. And we have how many classes? We
Sir,
have a Friday, Saturday, and one more. 3 classes we have. We will complete the course. We will complete the course as soon as, within our limit. I can do one thing if you agree, because your last class is on the 4th. So if you want, I can prepone it in one of the evenings. If you all agree, you let me know. I can send you a proposal.
okay,
I
sir.
can do that.
Sir, I'll ask it on the group and I'll
Yeah.
mail. Yeah.
Let me know so that at least you will get a couple of days. Your exam is, I think, on 6th. 4th is your last class.
Sir,
And
8 I think is the exam. Sir,
yeah,
BBL131 is on 8th.
it is on 8th. Okay, that is fine. So if it is on 8th, if you need 1 or 2, and exam is starting on 6th.
Yes, sir.
Okay, okay, that is what was in my mind. Your exam is starting from 6th. So if you want, that I can do. Second thing I can do is that I can take my second quiz after announcement, if you agree for the evening class.
Okay, sir.
So these two things I will write to you, and once it is agreeable, then only I will do it, because I have already announced that it will be in the class. What I find is that people, they are, because there are not many classes left, so we can do that. Okay then.
Thank you,
Thank
sir.
you. Thanks. Bye.